Good morning, everyone. Thank you for joining the webinar today. I see some more people are coming in, but in the interest of time, I definitely want to maximize everyone's time today, so let's get started. My name is Alison Almeida. I'm the project manager of the Gastric Cancer Registry, and I'm part of Hanlee Ji's lab at Stanford. So we hope this is really informative for you today, and we have a brief introduction from Alice Long from the Gastric Cancer Foundation. So she will do the introduction, and then I'll hand it off to Hanlee. Throughout the webinar, if you have questions, you can use the Q&A function at the bottom of your screen. I'll answer what I can in real time, and then we'll save a few questions for the end for a live Q&A session with Hanlee. And if we miss any questions, we'll be sure to record them and email them back to you. So, all right. Alice, I'll hand it off to you.
Thank you.
Thank you for joining, everyone.
Hello. Hello, I'm Alice Long. I am a board member of the Gastric Cancer Foundation and also a past co-chair. I want to welcome everybody for your coming to this event today. Today, we want to talk about the Gastric Cancer Registry and how it has been established, its current status, and what we are hoping to do in the future. So perhaps I can give you first a little background of how it came into place. And Hanlee, if you would advance to my first slide, please. The Gastric Cancer Foundation was founded by J.P. Gallagher, a patient. J.P. has left us many years ago, but the pioneering work that he did with colleagues identified that there is a big need for a comprehensive registry to support research for gastric cancer. In 2009, there was very little that was available to patients. So in 2011, the Gastric Cancer Foundation established the Gastric Cancer Registry in the Jim Ford lab at the Stanford School of Medicine. And we are forever grateful because Ron and Diane Weintraub have provided very critical funding to this effort in memory of their daughter, Beth Weintraub Schoenfeld. So the Gastric Cancer Registry was launched, and this is a HIPAA compliant registry. So the registry takes patient information, clinical records, tumor and saliva samples from patients, and saliva samples from immediate family, and put them all together to provide a comprehensive look in the hope that we can use the data to find more. Next slide, please. In 2017, the work was transitioned to Hanlee Ji's lab at Stanford. And the outreach to patient family is continuous. And I'm excited to let you know that we have also established new collaborations with large treatment centers with the hope to accelerate the pace of growth for the registry. Hanlee and his colleagues have created the Genome Explorer to ensure that all interested parties will have free access to this data. The hope is that no matter where a researcher is, he or she can tap into this data that provides support for their research. We are estimating that about 275 to 300 samples will be in the Genome Explorer data before the end of 2021. And we are looking at the potential, we have the potential to include another 200 samples from MD Anderson Cancer Center, but we are pending, of course, sufficient funding. So today, the webinar is the first opportunity for all of us to look at the progress we have made so far, and also inviting you and your colleagues to see how you might use it in the future and support its continued growth. Before I hand this over to Hanlee, I want to, on behalf of the Gastric Cancer Foundation, thank our collaborators, our supporters, and also our corporate sponsors who have been with us all these years to make all this work possible. Thank you very much for being here today, and look forward to talking to you more in the future. And Hanlee, it's yours.
Okay, well, thank you for that wonderful introduction, Alice. And I'd just like to have the opportunity to thank everyone who's taken the time to view the webinar. So just as an overview regarding the talk, we'll have a brief review of gastric cancer origins and challenges, a little bit regarding the development of the Stanford Gastric Cancer Registry, aspects regarding the Gastric Cancer Registry Genome Explorer features, including a live demo, and then future directions that are opportunities for collaboration and expansion of what we believe will be a very valuable data set that has clinical ramifications. So gastric cancer, as Alice has already alluded to, is really a major health issue. Internationally, there's 1 million cases plus that are diagnosed every year. It's a leading cause of cancer morbidity and mortality worldwide. And although there's been the assumption that the US is not as greatly affected as places such as Asia or Central America or South America, it's still a very devastating disease with fairly high incidence. So for example, 116 cases were diagnosed in 2017, and overall, the overall survival rate is extraordinarily poor, regardless of whether you're being treated in countries that are very familiar with this, like Asia or United States, with a general US survival rate of 32% over 5 years, much worse in some other countries that don't have the benefit of our type of resources. So here's a general estimate provided by the World Health Organization that shows the general distribution of gastric cancer. As noted, Asia is quite heavily represented, as well as South America, parts of Africa. But as noted, it's really an international disease, and it hasn't prevented that from any part of the world from being affected, including the United States, where it is actually a significant cause of mortality among cancer patients here. In part, sometimes it's recognized less because we have the disadvantage of not having the kind of screening protocols available that have been established in high incidences countries like Japan and South Korea. Recognizing it, there are sort of 2 general pathology types of gastric cancer, breaking it down into its largest categories. One is referred to as intestinal, which has some similarities based upon its histopathologic appearance to colon cancer, and the other is diffuse. I'm just noting this because this diffuse subtype, which is actually quite different in its presentation, oftentimes appears as a condition called linitis aplastica, and you see an example of that, where you don't see a gross mass that protrudes into the stomach, but rather sheets of cancer cells invade. And oftentimes this diffuse type of gastric cancer, stomach cancer as it were, is characterized by a very specific cell type called a signet cell. And you can see an example on the right-hand slide with the red arrow pointing to it. Notably, this is a type of cancer in the United States that seems to be not decreasing. And as I've had many clinical experiences with, this is a very devastating form of stomach cancer and extremely hard to treat. And oftentimes it's spread to other parts and other organs in the body once they reach an oncologist. Gastric cancer is caused by a variety of factors, and we're still working a lot of it. So we know that primarily they're environmental factors, such as a bacteria called Helicobacter pylori that are responsible for this disease. Other types of causes include infection called Epstein-Barr virus that's responsible for mononucleosis. High alcohol consumption, smoking, and dietary factors also contribute. We know that there are some general genetic risk factors, including very strong risk factors where there are families that have very high incidence of gastric cancer. This is related to a genetic condition called hereditary gastric cancer, and is typically caused by specific changes in a gene, or also linked to a type of colon cancer hereditary syndrome. However, the vast majority of stomach cancers are what we call sporadic, meaning that there's a variety of different features and causes that lead to how they arise and some of their molecular features. Notably, over the last decade, there's been a tremendous amount of work done in terms of defining what's known as molecular subtypes. So beyond these gross histology and pathology criteria, we very much know much more about the essential basis of some of these cancer types and what classifies one stomach cancer and differentiates it from another. So among this, studies, particularly from the Cancer Genome Atlas, as well as some studies based in Asia and whatnot, identified 4 major categories, including one which is called chromosome instability. This is where the genetic code as shown in large encyclopedic volumes that we refer to as chromosomes, are significantly deranged. Microsatellite instability, which is a result of very small genetic errors, mutations occurring in specific tracts called microsatellites. Epstein-Barr virus is a causative agent and leads to changes within what we term the epigenetic state of the cancer genome, specifically related to methylation. And then the diffuse gastric type that I alluded to is related to, or generally classified as being what we call chromosome stable, and has explicit genetic causes related to the exact same type of gene that leads to some of the familial hereditary gastric cancers that we see. As a GI oncologist, I've always found treating gastric cancer, especially metastatic gastric cancer, inordinately challenging. There truly are limited therapeutic options. Treatment still remains generally chemotherapy, although we have seen the emergence of at least several different types of targeted therapy, including ones that target HER2, which is also found in breast cancer and have been quite successful. Unfortunately, the vast proportion of gastric cancers do not have this particular molecular target that can be used. Other types of treatment that are based upon molecular changes within the stomach cancer itself include things called VEGF, which is a type of growth factor that stimulates the proliferation of blood cells, and more recently a gene called NTRK, in which specific genetic alterations lead towards this gene becoming hyperfunctional. And as a result, if you interfere with the function of this particular gene and this protein product, you can actually have an impact on stomach cancers. However, most of the targets I've mentioned are relatively rare. Excitingly, we've had significant progress in developing new types of immunotherapy for gastric cancer, particularly the one related to what I termed microsatellite instability. Explaining this type of molecular subtype of stomach cancer, in these types of cancers, what's happened is that there's a particular mechanism to remove genetic errors. Genetic errors we refer to as mutations, for those of you who are more laymen and not biomedical researchers necessarily. This leads towards a real susceptibility and an innate susceptibility to the patient's own immune system, and one can stimulate that with a type of drug called immune checkpoint. There's other indicators that are very similar towards this in terms of stomach cancer, and thus the use of immunotherapy is increasingly being recognized, but only a small proportion of patients have this microsatellite instability feature that allows them to actually benefit from immunotherapy. And so generally for metastatic gastric cancer patients, the vast majority will not have any benefit from this very exciting type of immune therapy modality that's increasingly being used in other diseases such as lung cancer. So what is the motivation behind this Gastric Cancer Registry and its use? Well, for one is that when we originally started this project, there were only a handful of independent data sets with the kind of comprehensive molecular and genomic information and associated clinical metrics. And so the founder of this, Mr. Gallagher, was very determined that this is a situation that needed to improve. Another major motivation is that there are really very few places where you can find gastric cancer where the data is centralized in kind of a clearinghouse with associated clinical data as well. And that proves to be very useful when you want to consider whether a target is something is a new target or whether there's a particular marker that might prove to be useful. Oftentimes the data is scattered, fragmented across multiple sites or multiple institutions or centers. So the real goal here is to try to centralize that. I think what's important, particularly regarding our own data set internally, as well as some of the public data sets that we have obtained, is that we have actually the raw data available. So we're able to expand the number of molecular, genomic, genetic features, even cellular features of gastric cancer by going and using new type of analytical methods on this because we control the data. So that allows us a couple of advantages, is that if there are new features we want to do, we have the ability to set up pipelines to analyze the primary data coming from our own tumor collections, as well as certain other public data sets, and subject them to the exact type and same type of computational analysis. And that allows us to rapidly expand informative features that can be useful for other biomedical researchers, release it in a way that's consistent, that's been quality controlled. In addition, one of the major goals and objectives of the Gastric Cancer Registry is identification of germline variants that contribute to risk. Albeit I did describe 2 genetic conditions in which there are diagnostic tests available, a substantial portion of patients who have a family history of stomach cancer do not have a recognizable hereditary risk, albeit they may have a family history that's very suggestive of it. So we are steadily accruing families and individuals who we can subsequently go back and determine if there is some type of unusual genetic variation that was passed to them from their mother or father that increased their risk of stomach cancer. Similarly, if a group has an interest in validating or discovering new clinical biomarkers, this provides an opportunity for them to have an independent data set they can go to verify whether they have something that can be reproduced. And so it's truly useful to have this independent data set just to facilitate and accelerate discovery of these important clinical biomarkers. And likewise, if groups have interest in identifying new cancer therapeutic targets and want to evaluate them, similarly, they can use this data set to go and do that. So the entire motivation ultimately is to accelerate the discovery and validation of these markers that may lead to new informative clinical diagnostics and also accelerate development of new therapies. Okay, so a couple of major goals that we have. As I mentioned, we want to really help researchers, clinicians better understand stomach cancer and really serve as a clinical scientific resource, a general open platform that will allow other investigators to develop new strategies for prevention, diagnosis, and treatments, as I noted. I haven't talked so much about prevention, but I'll talk about that and allude to what's happening in that area pretty soon. So we are funded by the Gastric Cancer Foundation. Their generous support has really enabled this project to occur, and they've shown the dedication over an extended period of time that has allowed us to really grow it. The initial development of the Stanford Gastric Cancer Registry involved a registry database that included family history, social history, demographics, and samples that would be later available for genomic data. And more recently, under our development over the last several years, we only took this project on several years ago, we've really expanded this and included new items such as considering individuals with gastric cancer and starting to think about developing and identifying families where we can go back and identify other types of mutations, consider also those individuals who have no mutations as well and have them be involved and provide clinical information about patients who are affected with this increased risk. We have an integrated team of molecular biologists, data scientists, bioinformaticians, and genomic researchers, all based at Stanford University. We have collaborations as well with a number of other investigators internationally who add additional tools, provide us with the ability to extend what we do, and that's been very invaluable for us. And the other advantage is that the entire team is very well integrated. They're proximal to each other. This really facilitates communication in order to achieve new goals in our research and improving the Gastric Cancer Registry. The registry site has been generally available. There was a revamp several years ago. It's accessible, and patients, this is oftentimes their first stop when they want to consider whether they want to enroll or not. So how does the registration workflow proceed? Well, first of all, a patient undergoes registration after they've received information. We use a variety of platforms in order to inform the public of the existence of this. The Gastric Cancer Foundation is perhaps our biggest advocate and enables a lot of patients to find us because we're fundamentally linked to the hip to the Gastric Cancer Foundation. We also conduct social media methods in order to do patient outreach, as well as enrolling patients and contacting them within the Stanford hospital and clinic healthcare system. So once this happens, the patient consents, they undergo a questionnaire for us to be able to determine more information from them, and then they authorize the release of gastric tumor tissues if they're an affected individual, or they can offer or simultaneously donate a saliva sample and return the complete sample kit. We've chosen to use saliva because it's much, much easier to obtain these samples in terms of obtaining normal DNA, particularly for those families who might have a hereditary risk of stomach cancer. And this has facilitated us being able to identify families and consider genetic studies in the future by switching over to this rather than blood. All of this information, as Alice alluded to, is maintained in a PHI secure REDCap database that's maintained at Stanford University. per all required regulations that are needed for this type of information. The patients see this very friendly online registration process and essentially are walked through that. They also understand the general intent is for us to develop the registry, not only with clinical metrics and information that are relevant for biomedical discovery, but for those individuals who are interested, they can actually contribute their own pathology samples. And through our dedicated staff and our program manager and Allison, we actually interact with a variety of hospitals to obtain these samples, bank them, and then subsequently catalog the information that comes from them in preparation for future genomic and molecular studies. Currently, the registry data covers over 412 fields. That includes participant demographics, medical history, family cancer history, and lifestyle behaviors. We are making efforts to expand this to consider whether we can actually obtain clinical outcomes. We have some of this data available, particularly from some of our future efforts for expanding out our cohort. And this is obviously an item that we'd like to continue developing, just so we've improved the utility of the data set for biomedical researchers everywhere. Currently, we have enrollment of 528. This is static because we always seem to be growing this number fairly quickly. And you can see based upon this Venn diagram, the overall distribution of patients, of which the vast majority are affected individuals with gastric cancer. A subset are those individuals who have germline CDH1 mutations and or a family history. And then extended pedigrees or families with individuals who aren't necessarily been diagnosed with gastric cancer, but are willing to contribute samples as part of the evaluation of their families as to whether there's a hereditary risk of gastric cancer or not. Our distribution is quite wide. We capture patients throughout the United States. As you can see, multiple sites. There's no particular region that's not been have participants who are willing to get involved in supporting our registry, and we're eternally grateful to these individuals who decide to consent and participate. In addition, we're making significant strides towards developing international registries, and I'll touch upon that a little bit later. Our current representation is listed here among patients with gastric cancer. This is not completely covering everybody. There is a bias currently towards Caucasians, but in Allison in particular, working with a number of other individuals, we'll have the opportunity to expand it, particularly regarding Hispanic individuals, in which gastric cancer is actually quite a significant problem. Likewise, we have ongoing collaborations with investigators in South Korea. And as soon as our first phase 1 expansion and data expansion in terms of our genomic analysis occurs, that's another item that we intend to get and pursue. Been slowed down a little bit by the COVID pandemic, but we continue to remain in contact with our collaborators overseas. Interestingly enough, we've also identified the general risk factors, and not surprisingly, Helicobacter pylori, listed as H. pylori, is one of the most predominant factors involved. We actually have ongoing research to better understand how this particular bacteria increases the risk of developing types of lesions that become invasive stomach cancer later. But you can immediately see is that we're capturing a fairly broad swath of patients who have a variety of causes or risk factors that they report. So this is a type of information which will be available for external investigators. In addition, when I first met the founder of the Gastric Cancer Registry, he was very determined that more information would be of any benefit. So major focus since the very beginning was to develop an ability to look at the genomic, molecular, and even cellular features of these tumors. And so as we've taken over the project and developed this through the efforts of my team, we developed what we call the GCR Genome Explorer. We're releasing this now, it's available, and at least in this first version, one can explore multiple types of genomic features in order to see what's available, and there's ways to download this data as well. And we'll take a look at this a little bit later, but I'm going to tell you a little bit about the details here. So notably, all the clinical parameters can be identified, at least the ones that we have consistent data on. And so, for example, you can see gender, ethnicity, cancer site, age of diagnosis, other types of health risk factors and whatnot. One important aspect about this entire project is the fact that there's an enormous advantage towards having the samples available and generating our own data. So one major item that facilitates the back end and makes this project feasible is that it's all managed very rigorously, including the use of the laboratory information management system. So this is really the back end that allows us to scale our study up to potentially a point where we could have thousands of samples and have this data available for investigators to look at. So we have the infrastructure in place for us to be able to scale up quite dramatically when the time comes. The genomic analysis production system consists out of integrating tumor tissue sections, pathology reports, clinical data sets, processing them for undergoing genome sequencing, so we can look at specific cancer features. We prepare what's known as sequencing libraries, and then what we do is really a multi-omic approach, which I'll describe later. And then we have automated bioinformatic pipelines that process this data and then subsequently generate some of the reports, which are compiled, that subsequently go into the Genome Explorer that are available for others to look at. This process includes obtaining tumor tissues. And then because we do something that's called multi-omic studies on these tumors, we extract two types of different nucleic acid analytes, RNA and DNA simultaneously, and use that in order to do method referred to as genome sequencing for those of you who are not scientists. This is a method of essentially looking at the genetic catalog of alterations that are specific to the cancer. For those of you who already know quite a bit about this, our multi-omic feature includes RNA sequencing, whole exome sequencing to identify mutations, and we use that for other purposes, as well as lower fold whole genome coverage that gives us very precise estimates regarding changes in the overall copy number status of these tumors. From this, we generate a multi-omic complete molecular landscape of these individual tumors. Again, one of the most critical things and advantages of the project is we have all the data, so we're able to do things with them that other projects and other registries are not able to do. And that's the real opportunity. So as part of the initial processing that I just cited, there's a first tier where we look at those fundamental somatic cancer ulcerations, the things that are very specific to the tumor cells themselves, that there were essential drivers that took a normal stomach cell and turned it into a cancer cell. And so that includes things that are called mutations, copy number alterations, gene expression. This is the multi-omic data set I was referring to. It represents different type of molecular, different type of genomic features that are found per an individual's tumor. We have access to all of these. And because we have the primary data, this allows us not only to look at these fundamental changes which led to the development of the stomach cancer, but it's also an opportunity to look at some very sophisticated other features that actually impact nature of how the cancer behaves and its cellular composition, and even how the immune system interacts with these cancers, as I'll describe later. So as noted, we have very automated bioinformatic systems. Literally every single process that I mentioned is in an automated bioinformatic processing step. So we control this process very carefully. We know exactly how the data is processed. And as a result, if there is some new innovation that allows us to extract additional information from these tumors, we can reconfigure our pipelines and our data processing so we can cull out that additional information later. And really, it's the fact that we have this consistency that I think will ultimately make our registry very valuable for the outside community. Now, importantly, and as I've been alluding to, there are a whole variety of different features that we're able to access. In part, what we're benefiting from are developments in my labs and many others across the world, where we can take this fundamental information from DNA and RNA and extract out a lot more features regarding specific stomach cancer. And these features are actually fundamentally important to the behavior of these cancers and represent opportunities to develop new types of biomarkers that could lead to improved diagnostics, as well as test ideas that could lead to novel types of therapy. So among these, what I term second-tier features, they're geared towards more specific cellular features. We're not just looking at discrete changes within genetic code, within the genomic DNA, within the gene expression itself, but we're extrapolating that information to tell us much, much more about how these cells are organized and how they interact, whether they have potential to interact with the immune system, and even the possibility of developing cancer vaccines, as I'll describe later. Furthermore, we can use the same information to actually determine if there are things like microbes, such as bacterial species, that have somehow been altered as part of this tumor cellular environment that exists within the stomach that is different from normal tissue and try to extract how these bacterial species may have an impact on development and maintenance of these stomach cancers in patients. That comes from all of this data, and it shows how we're able to really leverage sort of the centralized location and storage of this information, the consistent processing to continue to add value to the registry. So here's one exciting example where we do what's termed whole genome sequencing of a given stomach cancer. These are 2 individual patient tumors. And so what you're seeing is, in essence, an entire profile across the entire genome code, all the human genetic codes now available and visible. For those of you who are familiar with this, this is usually whole genome sequencing conducted at a coverage of about 5x or so, and allows us to see very explicit details of the genetic alterations that are fundamental drivers to gastric cancer. For those of you who are cancer biologists, this allows us to readily detect specific amplifications and deletions, as well as assess the overall genomic instability state, identify aneuploidy events, other critical features to determine what the molecular subtype of this specific stomach cancer is. So here's an exciting example where the blue arrow on the top showed an amplification of HER2, a gene called ERBB2, generally referred to as HER2 among cancer biologists. This represents a target for an antibody that's typically used as routine treatment for breast cancer, and individuals with gastric cancer can also receive this now with some significant therapeutic benefit. We can find other unusual features such as amplification of this gene KRAS. KRAS is another gene, another genetic driver of gastric cancer. And we saw a very unusual example where this particular gene seems to be amplified and thus probably has more function happening in the tumor cell inappropriately compared to the normal stomach cell. These are all opportunities to really determine, at least at a lower tier, whether there's a possibility of being able to identify and develop personalized therapies for these patients. This is the summary of the data within our registry, where we're looking across all the patients where we have tumors from the Gastric Cancer Registry itself, and then compiling this information. And this is what's organized within our Genome Explorer. So the red and the blue represent rather deletions versus amplifications, and thus one can compile all the changes that are present. So you can see as our numbers grow, this becomes more and more informative because it becomes very clear as to what the most common events are among gastric cancers. Similarly, we can look at these very specific genetic errors called point mutations. This represents some of the information that's derived from exome sequencing, for those of you who are familiar with these sequencing methods. And the upper columns show individual patients, and specific mutations are annotated off on the column located on the y-axis with the individual gene names. And so you can get a sense of the type of genetic errors that are present among these individuals in their tumors, particularly, that led to the tumor developing in the first place. And so with the Genome Explorer, you have the ability to go in and do much deeper searches. So this is an example where we're looking at a particular gene called P53. This is a particular part of the genetic code that's responsible for maintaining the integrity of the genetic code, referred to as the genome. For those of you who are familiar with this, you're all well aware of the fact that P53 is considered to be the guardian of the genome. And if you lose function, you tend to develop aneuploidy features and a lot of extensive genomic instability, copy number changes and whatnot that really lead to this chromosome unstable feature of stomach cancer. So this is a type of information one can immediately identify using our Explorer system. Similarly, we can actually look at gene expression from these samples, because as part of it, every sample undergoes RNA sequencing when the quality of the RNA is adequate. And so amongst our earlier pilot sets, as we're developing the protocols to do this off of archival samples, we found that we had very good information coming in from gene expression. Gene expression is really the intermediate code, for those of you who aren't familiar with this term, where it represents an intermediate that allows that particular genetic code to function and be turned on into a specific protein. And that's really the basic molecule within the cell that runs the function. And so this RNA represents an intermediate. For those of you familiar with the RNA sequencing and gene expression studies in cancer, you can see that we produce a variety of different molecular subtypes become immediately apparent based upon the gene expression profiles. And that allows us to make comparisons and also determination about molecular subtypes, as well as some very other critical features that are extrapolated based upon these patterns, as I'll describe later. So one of the major features that really helps us is that there are available data sets that have similar sets of information. And the most notable one was the study conducted by the National Institute of Health's National Cancer Institute, called the Cancer Genome Atlas Project. This was a multi-million-dollar project, enormous budget, in which several hundred stomach cancers were among the mix that was produced. What's really notable is that the first pass at generating this data set, which happened about 6 or 7 years ago, that cost literally probably $100 million to reach the stage. But because of advances in technology, that cost has been dramatically reduced. But that data exists, so we don't want to necessarily leave it there aside. And so as part of our development of the Genome Explorer, we're including that information so people can assess our data. And then from the Cancer Genome Atlas, they can see if the extrapolations they made in our independent data set are also correlated or confirmed in the Cancer Genome Atlas data set as well. And so this is the nature of where having multiple independent data samples available with the primary data becomes really useful, because you can go and actually verify across much larger populations and independent studies whether a particular genomic, cellular, molecular feature that you think is important in the disease can be verified in another independent study. More recently, we're starting to make headway in terms of leveraging the data available from the International Cancer Genome Consortium, which is a much larger entity. The data from that source tends to be a little more patchy and is not quite as comprehensive as what we're generating in-house per given sample, but nevertheless, it represents another important source of data that investigators can use with our independent data set, as well as Cancer Genome Atlas, in order to make verification and validation of particular aspects of the biology that may prove to be useful for a variety of clinical applications. So what are the kinds of things that we can do with the data that are really unique to this project because we have everything? So one example is that, as it turns out, most cancers are actually very heterogeneous in their overall structure. So you may have a tumor cell in one part of the tumor that is actually, in terms of its genetics, quite different than another portion. And so using the data we have available, we have pipelines in which we can determine what we call this clonal variation, these actual differences that exist within a single individual primary tumor, and determine some of those features. This is really important because this can be the basis of different types of clinical behavior and therapeutic resistance in stomach cancer. And so here's an example of 146 cancer samples where we can define this. Each one of these columns represent an individual stomach cancer, and the little colored bars that you're seeing represent portions of the tumor cells within an individual in which the stomach cancer is different. So it has fundamentally different genetic properties. And this can mean that these particular tumors that are very diverse, if you hit them with a particular therapeutic, they may behave very differently, and a portion of those cells, because they have different genetic properties, may not even respond to this therapy. So this is the kind of information you cannot see in most of the available studies, but we have it all centralized. And it's kind of information we intend to release that anyone can look at and use for their own studies afterwards. Using the same information, we can actually help and provide whether that particular tumor cell has produced something called neoantigen. Now, this is very important because the body's actually, and our own immune system is actually geared up towards fighting cancer, because cancers produce very unusual properties that the immune system should recognize, and the immune system actually may have the ability to attack that. Cancers are clever, though, and they figured out ways to dampen that, and thus basically prevent the patient's own immune system from attacking them. Nevertheless, these cancers have the possibility of activating the immune system through these neoantigens or neopeptides. And using the information that we have, we can actually derive this information and find out whether these particular neoantigens that are unique to the tumor and not found in normal cells can be exploited. So as part of our process, again, we have a pipeline in which we can generate and take the data that we generate from individual samples and produce this information. And so this is just a demonstration of that. I'm not going to go into the details. It's a little bit technical, but that information is all available. The information we also derive from this includes the fundamental genetics that an individual has that allows them to respond to any type of disease. And this is referred to as the histocompatibility locus. This is a very important component that determines how well a patient's immune system will respond to certain types of infections and whatnot. It also is a critical part to determining whether a cancer can be particularly targeted because it produces something unique to it, this neoantigen that's different than other normal cells. And based upon these features that are intrinsic to the patient, we can make predictions about whether there's any possibility that one can stimulate the immune system against the patient's own tumor. And so for those of you who know a bit about this and identification of neoantigens, which are the basis, for example, of the RNA cancer vaccines that other companies are producing, like Moderna, we're able to generate these lists and we provide them where these neoantigens, these fundamental drivers that would potentially allow a patient's own immune system to recognize these tumor cells. All of this information is available. There's also some information that allows you to determine whether it's present in the entire tumor versus a portion of the tumor cells. And so this is the type of information where we've leveraged our automated pipeline systems to be able to look at hundreds of these tumors. And so here's an example where we're looking at the different molecular subtypes demonstrated in this pilot study. And you're seeing, based upon this circle plot, the proportion of the individual molecular subtypes for the stomach cancer broken down. And then what we do is we can go back and look at that, this molecular subtype, and see how many of these candidate neoantigens are across the sample set. So for example, this represents, I think, microsatellite instability, which has a lot of different neoantigens present that are derived from these cancer mutations versus the other types. I think this is EBV and this is CIN. So this is the type of information that's released as part of our registry and our Genome Explorer. So we can do this across a variety of things. And I think the point I'm trying to make is that because we have this clinical information, this pathology information, and we have the primary data files that are managed as part of our system, we can generate this type of information very quickly. And for those of you who are familiar with this, this is the type of thing that can be used to verify neoantigen burden, which might represent a better proxy than, say, tumor mutation burden. Likewise, it provides some degree of inference that one can use to consider whether a cancer vaccine approach might be feasible amongst different sets of tumors. Now, one of the most important things that's happened, particularly with the development and the approval of these immunotherapies, for those of you who are not familiar with this term, this is where you receive a drug in which the patient's own immune system is stimulated to fight against the tumor itself, and has been quite successful in a number of examples. There are cases, particularly in stomach cancer, with this microsatellite instability feature, where a similar response can be seen. Well, it turns out that what's really driving that in the way these drugs work, as well as the way that the immune system is suppressed by tumors, you can actually look at the environment, the cell environment around a given tumor, by looking at discrete features of how those immune cells are arranged and the number. This can give you a sense about whether the tumor has been effective in essentially dampening down the patient's own immune system, or whether there's a possibility of stimulating it using one of these new types of immunotherapies like immune checkpoint that would give you the opportunity to be able to intervene with this type of therapeutic strategy. So what you're seeing here is a stomach cancer, and then in closer resolution of this panel is the use of multi-colored immunohistochemistry strategies that allow us to pick out the individual immune cell types. There are a lot of different immune cells, for those of you who are not familiar with this term. For those of you who study the tumor microenvironment, this includes CD4, CD8s, Tregs, whatnot, and we can determine the relative density, the proximity to tumors, to the tumor cells within a gastric tumor. Well, it turns out that you can actually infer some of this data using a variety of different methods. So one of the most popular methods, albeit quite costly to do, is to actually sequence out all of the individual cells from a given tumor. And this is an example from one of our studies where you can take a stomach tumor, break it into its individual cell components, and then sequence all the individual parts, and then look at what those cells are. However, there are ways to do this from the data that we have. So you can take these RNA-seq files, this gene expression sequencing data, and you can look at the actual proxy estimates regarding the type of immune cells that surround that tumor. And this is type of data that's available as part of the registry. So just to give you a sense about what this looks like, I have a slide, but I'm also going to take a little break here and do a bit of sharing to show you what it looks like. So if you don't mind, I'm going to modify my share just to make give you the opportunity to take a look at what the actual Explorer looks like. So you should all be seeing my web page, and this represents the GCR Genome Explorer as it stands now. You can see a variety of features. You can select all. You can select these different populations, including the TCGA or our own sample sets from our pilot study. And then you can look at different features. So here's an example where you can see the individual breakdown, age of diagnoses. This is from our own gastric cancer. This is actually from the TCGA study. So you can see the number of patients, their age representation, their survival, cancer sites, gender, ethnicity, whatnot, tumor stage. You can look at the type of mutations that exist across this. You can also look at gene amplifications and deletions that occur off of this data, which can also be downloaded as flat files. Here's an example where very useful information about the patient's immunogenetics, particularly regarding these HLA genetic alleles that exist. You can see a breakdown of this. You can individually look at the number that exists across all of them. You can look at the immune cell composition across patients here. So these are the different cell types that exist, that are known immune cell types that are present within the tumor surrounding it. And you can see the general composition here. And you can also see the microbiome, where we're using the same RNA sequencing data to basically show you what kind of bacterial species are present. So here's another example from our own data and from our ongoing gastric cancer registry. This is part of the pilot study, which we used in order to help set up the Genome Explorer. The numbers are going to increase significantly. And likewise, you can see similar features here. And then you can go into greater detail. You can look at individual mutations. So here's an example of P53 where we can compare our study and the proportion of mutations compared to TCGA. So by having all of these integrated data sets in our own sets, you can essentially conduct studies where you may discover something in one data set, and then you can see if it's verified in an independent data set. And I think that's one of the advantages that our system provides. Okay, going back to my presentation. So one thing you can actually do with this now is you can take these different features and start to combine them. So this is where we're looking at these neoantigens, these particular genetic alterations that change components that are unique to tumor cells, and comparing them to the cellular context, particularly the immune cells that may be present around that, and use that as a way to gauge whether those neoantigens are somehow recruiting immune cells versus having immune cells that normally should be seeing them, but they're being turned off. So this is the type of data that's available for individual researchers to examine, regardless of context, and to consider whether there are opportunities to look at develop new biomarkers or therapeutics. Now, as I already alluded to, the stomach actually is not a sterile environment. There's fairly rich microbial species population that exists. We refer to this as the microbiome. And most notably, one type of bacteria, Helicobacter pylori, is probably the predominant risk factor for most stomach cancers. And so this is an example from one of our collaborators, Manuela Miva, who's an infectious disease doctor, who's indicating the presence of this Helicobacter pylori that's present in stomach tissue here. And after you take antibiotics, this is eliminated. So for those of you who may have been diagnosed as having Helicobacter pylori, you received antibiotics to eliminate these bacterial species and thus reduce your risk. Well, as I already mentioned, using this integrated data set that we have, we generated from our sample as well as from what was released 6 or 7 years ago as part of the Cancer Genome Atlas, we can derive very similar profiles about what kind of microbial species existed around the stomach tumor. And as I mentioned before, we have automated pipelines to generate this information, and as part of our Genome Explorer release. This data set will continue to grow. And because we control all the data, for us to add new features or make improvements in analysis or identify novel things as people make improvements in computational methods, that becomes very straightforward for us to do. So the GCR Genome Explorer is really living entity. As the field advances, we can leverage that and make the Explorer even more informative for our target audience, for those individual researchers or even other users, people who are just curious based upon having a diagnosis or knowing someone with a diagnosis, where they can go in and they can literally do science right at their own computer. And so here's an example of some of the samples that came in as part of our Gastric Cancer Registry, where we have multiple types of bacterial species that were noted around the particular tumor. And you can see the general breakdown based upon their genus as to what the composition was in terms of the bacterial species that are present around that tumor. And sometimes that shifts. And so that's an important feature because there's increasing information that suggests, and increasing science that suggests, that the features of the microbiome actually have a significant role in determining whether a patient can mount an immune response against a tumor, whether it's initially or whether they receive an immunotherapy. So again, valuable information that only proves to be useful if you put it and you can integrate this. And it really requires this integrative approach where you have the sample, you have the primary data from the sample, and you can extract all these additional features from it. And so what is the next phase? Well, I think importantly, we're in the midst, thanks to the generosity of the Gastric Cancer Foundation, in increasing the size of the cohort. So we have, in collaboration with Intermountain Healthcare, a major integrated healthcare system that covers the Mountain West area. We've obtained a large number of samples, and using this additional funding, we're going to be populating the Genome Explorer with this data over the coming months. So at the end, hopefully there'll be several hundred tumor samples available that people can explore, and that just makes it more useful as a large aggregation of data. It makes it more useful for improving the statistical power to validate and discover new findings from stomach cancer. And because we have the Cancer Genome Atlas also available in the same data set, people can almost immediately make comparisons between two independent populations and determine whether what they're looking at that they think is important can be verified elsewhere. Really a rich data source for conducting stomach cancer research overall. Future directions, I'm limited in time, so I'm not going to go into quite as much detail as I have slides for, but as already noted, the microenvironment in which these tumors exist, they're surrounded by a whole diverse types of cells. That actually determines an enormous amount as to whether that tumor is going to be responsive to therapy. And given all the excitement of immunotherapy, again, stimulating the patient's own immune system against this, there's much that we can derive and additional features we can derive in terms of the immunologic cellular context around these tumors. Finally, in collaboration with the Division of Gastroenterology and another registry study, we're now also getting involved in trying to develop preventive strategies, because ultimately prevention of stomach cancer is probably the best thing to do given how devastating it is once you have the diagnosis. So what I'm showing you now, an example of just the sheer complexity of looking at normal stomach tissue, as you see on the left, versus development of cancer where the cells become deranged and whatnot. As an added complexity beyond the fact that the tumor cells have taken on very weird forms and have all kinds of molecular and cellular aspects that have gone awry, the actual context of these tumors, the different immune cells and other types of supportive stromal cells that exist, influence how these tumors behave. And so as an added element for future development and opportunities for collaboration really is that we'd love to build out and include these cellular microenvironmental features beyond what we already have. And this would focus primarily on immunologic cellular features, given the importance of developing new types of immunotherapy. So my group specializes in single cell sequencing, and we've conducted pilot studies where we can look at individual cells. For those of you not familiar with this, what you do is you take a tumor and you break it down into its individual cell components, and thus you can separate normal cells by individual cells. And the way you do that is you sequence them out. So you're sequencing the individual genome contents of a given cell that comes from a patient's tumor. And we found this to be enormously useful and have identified that the cell types that are responsible for the stomach cancer, if you look at them in terms of precursors that aren't fully cancer versus the full cancer, we can actually see some very significant differences in terms of how those cells are constructed and how they're behaving based upon the single cell sequencing approach. And so that allows us to identify potential markers that can be used in a context which is a little less expensive and can be extended to thousands of samples where we can verify those types of cells, whether they're present or not in a given patient sample. So as our registry grows, we'll have the ability to use these types of microscopic staining methods to be able to analyze the presence of different markers that might indicate different types of cell states that exist among the cancer cells and the pre-malignant cells. Similarly, as I already mentioned, the immune cell context that surrounds a given tumor is a very important feature. So for example, for those of you familiar with immunologic tumor microenvironment features, this allows us to look at CD8, CD4s, natural killer cells, cytotoxic T cells, T helpers, T memory cells. Now, expanding single cell sequencing studies to larger cohorts is really cost prohibitive at this time, but there are other ways to do this. So another way includes using advanced immunohistochemistry panels that once we have particular markers that delineate some of the cell types of interest, such as Tregs and whatnot, we can use that as a very efficient way of determining amongst our cohort whether immune cells were proximal, the general density of specific immune cells around the tumor, and use that as an added layer of information that we add on top of the genome sequencing features that we describe. And so this is an area where I'd really love to have opportunity to collaborate, consider joint studies, particularly for our partners or future partners in biotech industry, where we can consider using our repository to provide this type of information and use this to help inform in development of new therapeutics, particularly in the immunotherapeutic space, but there are opportunities in other types of drugs as well. So you can see example where we can do this type of staining, multicolored staining, in order to identify these important immune cell types and their relative composition, and determine across a population the range of these features that exist across different gastric cancers. And I think is an opportunity to consider the opportunity within a preclinical context of a particular immunologic target. This is really required information that's I think will prove to be very useful. And certainly in terms of our own immunotherapeutics discovery targets that we're looking at and evaluating, this information becomes very critical to determine whether there are enough patients that have this target available that we can exploit subsequently. One of the things that really will make this much more feasible is the fact that we have very advanced imaging analytics that use machine learning and whatnot. So we have a manuscript that's currently under review where we can apply these methods to these very complex analyses using even spatial transcriptomic sequencing to be able to delineate these types of cell features that are found amongst our tumors. And again, because we have these samples readily available, we can add these layers of information to increase the utility, to increase the overall value of the registry and its Explorer. So I'm going to wrap this up by just talking about the last exciting part of it, is that through collaboration efforts with Dr. Juha Wang and Robert Wang, who's in the division of gastroenterology, both of these are gastroenterologists, we're now in the midst of developing and leveraging what we've learned in some of these methods towards understanding why some people go on to develop gastric cancer in the first place. And this is separate from the genetic component because there are individuals who develop these pre-malignant lesions, and we're trying to figure out why this is and how many of those individuals go on to develop stomach cancer. And so using many of the same type of methods that I've described, we're now working in conjunction with this GAPT group led by Juha and Robert to apply similar types of analyses to their pre-malignant precursors of stomach cancer. And I'm quite excited about this part of the project. So opportunities to collaboration, just as a wrap up, really, we're hoping to get through a couple of hundred samples and release that data as of 2021, much to the support of the Gastric Cancer Foundation. But because we have the infrastructure in place, we have the know-how, we have the informatic analytics, we have the computational methods, I think there's ability to expand this significantly. And numbers help. So there's a number of some of this information is available from Cancer Genome Atlas and whatnot, but having these studies powered to include more samples just improves the ability to make discoveries and simultaneously or rapidly validate them. We're working to increase the ethnic representation. So Allison has been very good about identifying places that treat populations with significant Latino population. Those individuals are actually in very high risk of developing stomach cancer, haven't really been represented in the current studies. We're going to address that in the future, but that's certainly an opportunity to collaborate. There are the immune cellular features of gastric cancer through these types of visualizations through advanced microscopy methods, as well as something called spatial transcriptomics. We can provide even more information to what we currently have that should be useful in determining what the immune state is or what the cellular microenvironment state around those tumors are. Through our own publications and our work through single cell sequencing, I get requests all the time to release our data. And I think the Gastric Cancer Registry Genome Portal provides an opportunity to organize our data as well as others in a centralized fashion so that we can release that and allow other groups to evaluate this as an aggregate. So rather than having a lot of people just try to put data together, that's a lot of wasted effort. Perhaps there are opportunities to get all that data together in a single place and release it and allow people to analyze it jointly. And then as you've seen, there's these novel integrative features that we can exploit to really improve the value of the data that really reveals features that we wouldn't even thought about, say, a couple years ago, like microbial species and whatnot. That's the advantage of having the data at our hands and at our feet, because we can immediately apply these novel types of analysis methods to our data sets and then release them. So again, another opportunity for collaboration, and hopefully one that would interest support both among our academic partners and our industry partners as well, who are working in this area. So contributors and collaborators, Allison Almeida, who's the program manager, has been absolutely instrumental in driving this program and making it what it is today. So much of our thanks goes to her, as well as Maddie McNara, who's been working with her for the last year. The two of them represent our dynamic duo, and they've really helped spur this forward. Then other members of my group contribute various parts of the data, including Stephanie Greer, Jay Chen, Ho-Jun Lee, and Nuja Sata, and Susan Grimes. Our collaborators in gastroenterology, we have a unique partnership with the current chief of surgical oncology, George Poultsides, who specializes in gastric cancer. Great collaboration with him. He's been very supportive of our efforts. So we're really linked at the hip with surgery here. And prior contributions from James Ford, Meredith Mills, Paul Van Humlen, Matt Cubitt, and Maya Archie for helping develop the registry and its inception. So that's our website. Feel free to visit. And much thanks again to the Gastric Cancer Foundation for their vision. We are following the marching orders of what Paul Gallagher told me at the very beginning, and we intend to get across the finish line and really achieve his aim of having a registry that's informative, not only in terms of clinical data, provides a wealth of genomic data that will facilitate and accelerate discoveries in this area. Thank you.
Thank you so much, Hanlee, and thanks to Alice and everyone who stuck around. I didn't see any open questions, but I did include my email in the chat box if anyone comes up with anything at the end, or if you are exploring the Genome Explorer yourself, go ahead and send us an email if you have any questions. This meeting will be recorded, so I'll be sure to send Stacey the link so she can distribute to your colleagues and yourself if you want to take a second look. But ultimately, we'll let everyone go. Thank you so much, Hanlee, and thanks again, everyone, for your time.